flew in to the conference. Nick had to go on. He got up and he gave his talk, and then I got up and I gave my talk after him. And then we took questions, and this guy kind of rolled into the questions, one of the panelists, but he hadn't been there for our talk, and we hadn't been there for his talk. And one of the people in the audience, it was a physician, and he raised his hand and he said, how do you guys come up with this stuff? And Josh said, I don't know, you just sort of throw something at the wall and see what sticks. And at that point, I think both Nick and I were kind of sold. We were like, oh, this guy is pretty interesting, right? Really interesting personality. And so we ended up, the panel kind of took on this, back and forth between Nick and Josh and I, as we learned about what he did, which, is really exciting and totally unprecedented. He's definitely a pioneer. And so, if we are sort of the Wild West in the field of neurostimulation, then Josh is out in the middle of nowhere where there's really no civilization, because this is a brand new technology, and he's one of the first people to really pioneer its use. So I'm looking forward to hear what he has to say today.
Hi, everyone. Tiff?
Yeah.
I just heard from Josh. He needs about 3 minutes.
Okay. I'm just wondering if there's anything in particular that we should mention that's coming up.
We do have the advanced training coming up here on May 6th through 8th in Denver. Tiff and Nick will be at the AAPB conference presenting and tabling. We do have next week a series of case studies. The case studies for beginners on Monday, ERP study group on Tuesday. So that's the 22nd, 23rd. And then the case studies for intermediate advanced on the 24th. We have special series coming up on the 2nd of May, the NeuroStim for clinical profiles. The topic that day for the month of May is anxiety and stress.
Oh, perfect.
Nick is offering, one of his Neurofield software webinars for Neurofield ERP on Friday, May 3rd. That's at 9 a.m. Coming up is another special series called the NeuroFam Hot Topics. That's on May 16th, Thursday morning, 8 to 9. Understanding independent component analysis. And that's followed the next day by Nick's Catch Up with Nick with his May messages. Beginning on May 17th, we have a brand new series, 10 sessions of EEG for beginners.
That one's going to be fun. And that's a weekly, is that right?
I believe there may be a week that's skipped, but it is 10 weeks starting May 17th, and it runs through July 19th. 10 sessions.
So the purpose of that, every course every day is a bit different. And topics like montaging, morphology, vigilance, artifact, a whole host of things are going to be covered. And the purpose of that series, I did this last year, and it's just to get people up to speed on the fundamentals of beginning to read EEG, because I wasn't taught it out the gate, and I sort of had to scrape and scrap together things that many of us on the call right now know very well. But it'll be a good one. I really look forward to that series.
Yeah, it was very, very popular last year, so that's a good one. I'd also like to mention that we are having a free public event in Santa Barbara on the evening of May 28th. That's a Tuesday from 6 to 8, where Nick and Tiff will discuss demystifying neurotherapy. So they'll be sharing their perspectives on the practice and implementation of neurotherapy, and sharing that with a pedestrian public so they can learn about how it can advance their own health. It'll be available live streaming as well as in person in public at the Unity Santa Barbara, which is here on Anacapa Street. And there will be information online and sent out to each of you through email and social media. So you'll be able to learn the details of that as it gets closer to. Oh, I was also going to mention that we've just launched early bird tickets for our annual conference. Conference is November 7th through 9th, and early bird tickets are available now at a $250 discount.
And then there's John LeMay's ERP course as well, which that's another one that I'm really excited about.
That's a very special one.
Okay. Hello.
Hey, John.
So the way that I'm going to be structuring this is that I'm going to attempt in this hour to cover the entire field of focus ultrasound something I've made myself do or try to do a few times over the years, but as we get going, it's like more and more stuff. So we'll see if I can do that. Talk about the history, the kind of present state of things, exactly, what the hell is this that I'm talking about, and then what I think the near future is, and then if we have extra time, I will get into kind of like really nitty-gritty scientific details, and we'll just get through that until I get cut off. So Tiff, you just maybe just like let me know. Okay, so I'm planning on just going until.
I'll give you a 5-minute warning, okay? At the
Okay, perfect, great. Okay. So so anyway, we got my intro and everything, but here's all my information, and information for my current job, this nonprofit Institute for Advanced Consciousness Studies, and some of our studies ongoing right now. Okay, so let's begin here. So, of course, focus ultrasound, what we're going to be talking about today for neuromodulation, is one of many different forms of accomplishing neuromodulation, right? Another word for brain stimulation. So that's anything that we use to alter brain activity. Some people consider even psychoactive drugs to be a form of neuromodulation, right? But focus ultrasound can specifically be a form of non-invasive neuromodulation. We can make the PowerPoint available. And these are more limited. There's the fewer types, increasingly many, that I'm now hearing about from people like Tiff here. With some of the most popular ones being TMS, transcranial magnetic stimulation, and TDCS, transcranial direct current stimulation. There's different words for that as well. Which, unlike things like deep brain stimulation on the left, right, where you physically put an electrode in someone's brain, can alter brain activity through the skull without causing any sort of tissue damage whatsoever. So you can do it in healthy people for things like basic science, in most use cases which don't justify a full neurosurgery, of course, right? So focus ultrasound is one of these methods, or it's joining these other methods to accomplish that non-invasively. I want to stress that it is a super new field in a sense. So in a little bit, I'll talk about the history. We've actually known for a while that ultrasound can induce neuromodulation, alter brain activity, but really just out of chance, it wasn't studied much until very recently. I guess that's relevant for me as well. So while I'm, a very young scientist, I'm one of the people that has used this technique the longest. It was really me and me and my PhD advisor at the time, and 2 other groups in the United States, 1 Korean group, all at exactly the same time, about 2015, 2017, began using this. So there was some growing interest here and pretty much just like 1 group. You did a bunch of early animal studies in the early 2000s. The first human use happened here in 2014-ish, right? So it's actually unclear exactly who did it. It was either one of the patients in the first clinical trial, or it was this guy Winley got out on the East Coast here. It's actually unclear. Okay, so that first clinical trial that I mentioned happened at UCLA, and while the first patient actually the procedure was performed, then in 2014 I kind of took that on in 2017 and ran it to completion, and I'm actually still publishing the last study associated with that. Okay, so I just want to go back to while like TMS remains much more widely used, of course, you can obviously see this exponential trend, right? And now if you view them overlaid, which I don't think I put here, it's focus ultrasound now starting to take on some at least visible part of the non-invasive neuromodulation space. Oh no. Okay, so what is this thing, right? I'm talking about. So what is focused ultrasound? It's exactly what it sounds like. It's ultrasound, just like you would use in ultrasound imaging, like in imaging a fetus, for example. It's just sound waves above the range of human hearing. Exact same technology, with some differences in frequency and that sort of thing. I have this image here that for some reason animated to disappear, but showing that the only difference between like imaging ultrasound and the ultrasound that I use for neurostimulation is that imaging ultrasound comes out of the transducer, out of the device that produces ultrasound in kind of like a flat wave. It just comes out, bounces off some stuff, right, in this cone pattern, comes back, you measure that, and that's how you get your image. Whereas focus ultrasound has a curved transducer, a curved element, right, such that it projects ultrasound in an inverted cone so that the ultrasound energy coalesces onto a particular focal point. In our case, inside the head, inside the brain. And this way, you can induce neuromodulation, brain stimulation in a particular little region inside the brain, right, depending on how you aim it, without doing so in the intervening tissue, right? So you project this energy through cortex, through the deeper cortex, right, maybe approaching the thalamus. It's an area that I've targeted a lot, right? And it reaches an intensity there and only there that's high enough to induce brain stimulation, altering activity in that region specifically. That's the basic concept. So this is an animation of exactly what I just said. It comes in, so it's exactly like a magnifying glass burning a piece of paper, right? Pretty much exactly like that. Okay. And so, just like I said, you get this focal point somewhere in the head. Now, the nature of using these single element transducers, we'll get into what that means, but just like a single source of ultrasound, which is what's typically used in science today, is that it makes it sort of elongated focus. So don't think of it as a single point, though that's possible. Think of it more like a little sausage, right? It's something like about half the size, maybe like a little smaller than the end of your pinky, right? Placed somewhere in the brain at any angle you desire, but in any part of the brain, even deep brain regions that are impossible to hit with TMS and TDCS. That is just like another view of the same thing. Okay. Now, okay, great. So interestingly, while science using focused ultrasound or neuroscience, right, that use it for neuromodulation, brain stimulation is extremely new. It's really now just hitting its stride, as you saw. Focused ultrasound has been used in medicine to ablate tumors and other little bits of brain tissue for now approaching a couple decades. But this is done a little bit differently. Instead of a single source of ultrasound, you may have hundreds, and the energy is projected out in such a way that it temporally coalesces, right? Like the peaks of the waves from each of these elements coalesce onto a single digitally aimed point inside the head. And so you can burn out a single 1 by 1 by 1 millimeter area inside the head. Maybe to treat Parkinson's or essential tremor, this sort of thing. It's amazing. You can have people awake during this and see their hands outside of the MRI, which is used to do targeting, shaking, and it'll just go away, right? Much like deep brain stimulation for the same reason. The reason we don't use this, I can talk about this more later, in science is because you need a CT. These machines are extremely expensive, and you need to be, it's not invasive, but you need to be really bolted in. It's not a comfortable experience. Yeah. Okay. So how I've been talking about this so far is really theoretical. This is what I'll bring in my lab here. But what exactly does this look like in the context of a modern scientific experiment? This, at least in the case of my equipment, using brain sonics devices, looks like this. Okay, so this is a single element transducer. Inside this casing, right, is just a curved element, or sometimes a flat element, but then there's some some materials in there that guide it into this cone shape as it comes out. And you can physically place this right anywhere on the skull, project through the skull and brain, and obtain that focus point that always just goes away that we're talking about here. So how do we leverage the, in theory, incredible precision that we'll talk more about in a real experiment, given that you can't peer inside the brain? We use some form of neuronavigation. So somebody has an MRI prior to an experiment, no radiation necessary. You can do this in any basic scientific experiment, and you use like an infrared camera like this, trackers that you place on the head to know where the subject's head is, register the MRI to that, and then in real time you can steer this thing, which also has a tracker on it, right, on the head and look exactly where the focus point is, in theory, being generated inside the brain. So you can accomplish pretty readily with this equipment, this technology developed for TMS, but adapted to focus ultrasound, approaching, something approaching to millimeter or at least like 1 centimeter range precision. Okay, so this is like what a modern focus ultrasound setup or lab looks like. This is at the Institute for Advanced Consciousness Studies in Santa Monica here. I'm pretty sure this is certainly one of the more professional, if not the most professional setups in the world. It's still such a new thing that while this is changing, you kind of have to almost make some of the equipment yourself with little 3D printed adapters like this. Of that nature, right, that help us use this equipment designed for TMS to do all of our targeting, sort of thing. Yeah, back in the day, this was super crazy. I basically made everything besides the camera and the software to make it work. Built a chair, whole chair, everything to like stabilize the head. But that's rapidly changing as the science progresses, or as the field is coming into the mainstream. So as you can see in the top right there, that's one of my colleagues, Leo, being set up in the chair where we did a meditation experiment as an example. And be warned if anyone actually wants to do this in scientific context today, right, barring some new technology built, is quite a laborious setup. So why do I go through all this, right? And use focus ultrasound as opposed to these other ready-made technologies that you can just buy equipment for? And the reason is ultrasound has incredible many benefits above and beyond those of these other technologies. I've kind of talked a little bit about this already. It's impossible to talk about this without going into the sum, but you can think of all these neurostimulatory technologies as existing on a plane, right, defined by the depth of their influence, like how far in the brain can we get, or what part of the brain can we target without there being some problem, and their spatial precision. So for example, TDCS, which just arcs electricity through the brain, can in theory hit pretty deep, if done the right way, but it has very little spatial precision. again, it depends on your techniques, and it's arguable and everything, right? But certainly until very recently, very little spatial precision. Sometimes you're arcing it through, like a whole big part, so maybe half of the left frontal cortex, or this sort of, depending on many factors. Okay. Whereas TMS, while it's more precise, while it's more precise a bit than TDCS, producing something like a 4-centimeter circle of influence on the very outside of the cortex, it can only hit that cortical mantle, just the very outside of the brain. So you can only modify, you only directly modify functions that are mediated by this very superficial part of the brain, and nothing else, unfortunately, unless you kind of do like this complicated network stuff that's only so effective. Where we want to be is where DBS is, where deep brain stimulation is, where you can target any brain region you want with incredible spatial precision. Though, of course, right, you need to do surgery for that. It's pretty close to where focused ultrasound is, especially when considering that deep brain stimulation, while it's really precise when you get it in there, it's actually usually off a little bit, just because of the nature of the surgery, to about the same degree where focused ultrasound is. Okay. So while there are, in theory, these advantages, of course, like any new scientific field, there are some hurdles. Maybe limitations are not even the right word anymore, because we're increasingly able to tackle these. I see them more as some projects that the field's working on, and we're getting pretty close to being able to deal with some of these. So the main limitation of focused ultrasound right now is the ability or the lack of our ability to completely predict how the skull affects the beam coming in. So you can design a transducer that perfectly produces a focal point in water, and this is what is done when you're testing it. But the second you put it on someone's skull, right, the variations within the skull shape and density, which actually vary a ton between individuals, entirely randomly, it's going to modify, mess up this focal point. And on top of that, there's like so many things that affect this, so it becomes really hard to predict, right? Like the thickness of bone, like kind of how the trajectory you have, the angle at which you're going into the head, the frequency you're using, the size of the transducer, right? All of these things. So it's not as simple as, okay, you got this thickness of bone, you go and run this formula, and there you go. Okay, so for example, look on the right here. This is just like some computation simulation that I ran in an old paper showing the difference between in water, right, and then through skull when I'm targeting the, in this case, the globus pallidus, so like the basal ganglia structure, through even the thinnest part of the human skull, which on average is this, the certain part of the temple, which we're trying to go through to minimize the skull refractory effects. But even so, instead of this nice little focal point, you get this kind of like smeared thing, right? I kind of, over the course of an entire experiment, it's kind of like probabilistic cloud, right, of influence, depending. And even sometimes it can be biased in one direction, right? So this is a big problem. Like I kind of said, we typically use acoustic simulations to account for this, but right now, there's only so much. Most people just run a simulation through just a reference CT before an experiment, say like we expect about this amount of attenuation, about this amount of refraction before doing it. And the individual variations in in real humans and real trajectories and real experimental conditions are going to limit that particular simulation. I'm going to just plug a little study that I did. It came out in 2021 in +1, where you can input all of those factors that I just brought up, and it'll spit out an estimate of how much we're reducing the intensity, which skull does, and how much on average we're expected to move the focal point itself over, right? Which I'm calling in this case refraction. You can see some outputs from that program that's publicly available for free right here, showing how much something like the frequency of the ultrasound itself, we call this like the fundamental frequency, affects the focal point, right? So if you have like a lower fundamental frequency, you have a longer wavelength, and so as these waves coalesce, right, in the focal point, it's only possible for them to generate a certain size of a focus. So on the right, where we're using a lower frequency, you get this larger focal point, right? On the left, higher frequency, smaller focal point, and there's some complexities there. Like when you use the higher frequency, it's harder to get through the skull. We could talk about all of that, but that's just one example here. So this is, just one simple way to easily, get an estimate of what to expect in a certain experimental condition, but it's not perfect. And later, if we get to it, I'll talk about, we'll definitely get to some of it. I'll talk about how we're beginning to deal with this in better and better ways. And it's going to be fully possible in the coming years, I think. So that's really the main limitation. The other one, really related, is the safety of focused ultrasound. There is some point at which obviously you can cause damage because it's used at high intensities, really high intensities, to burn out little parts of the brain. If you surpass this threshold, which is not fully known, but don't get too scared, you can obviously cause damage. And in order to, go up to areas that become a gray area in terms of safety, you need to be, in an individual, you need to be able to really precisely predict how much the skull is affecting focal properties in that person in particular. Okay. We know enough, I'll just say right now, we know enough to be very confident that, say, experiments that I run are fully safe. But there is a desire to maybe go higher, up to higher intensities that are beginning to be questionable, to get like larger effect sizes. Okay, so real quick, let's talk about the overall history here. So let's go back to our modern exponential curve up in how much attention focused ultrasound's gotten, or focused on ultrasound neuromodulation's gotten in recent years. Okay. And recognize that we have to go all the way back to the late 1950s to find when this effect was first recognized. So this guy named Fry at this time was working at the University of Illinois on acoustic technologies for the military, kind of caught up in World War II and immediate post-World War II, early Cold War era, like infinite funding, and did a bunch of crazy experiments using ultrasound and water and ultrasound weapons and medical technology and this sort of thing. And he noticed some strange behavior in some of his animal models, and he thought like maybe this can affect neural activity, something along these lines, right? And the first formal experiment that he did that demonstrated a reversible neuromodulatory effect. So I'm altering brain activity, but I'm not causing damage because it's not permanent. Was this one depicted down the bottom in a cat. So one of the earliest or simplest findings in neuroscience is you can flash a light to the eyes. It'll percolate through the visual system, hit the back of the brain or the visual cortex, and cause this big VEP, this visual evoked potential. So a really classic thing that he had access to at the time. In order for that signal to get all the way through to the cortical visual system and generate a VEP, it needs to go through the lateral geniculate nucleus, some small region deep in the brain that he could focus ultrasound onto, which he did, and see if it altered the VEP, and in fact it did, right? In just the seconds after ultrasound was put into the lateral geniculate nucleus, but not 30 minutes after, right? Showing this reversible effect. And he, of course, immediately recognized the potential benefits of that. On top of that, he actually also did the very foundational work using focused ultrasound for non-invasive medical procedures, exactly how it's mostly used today, right, to burn out tumors. And that's what you see on the right there. So he did very early work. But unfortunately, in 1968, like so many great people, he died, and so did the entire field of focused ultrasound. So how's the technique different than the TFUS techniques discussed and published in the web? Yeah. Okay. So it is. Yeah. Okay. So this is a good question. So how is this work different? How is this technique? I mean, this is the same technique, right? This is a very early version, and then this 2013 paper is kind of you could call that's kind of the beginning of modern work in humans, but it wasn't technically focused. Actually, they just took an imaging ultrasound transducer, maybe turned it all the way up, and then put it on the frontal cortex and blasted ultrasounds into the frontal cortex, right? I think later they maybe used a focus transducer in some people, but this was just an imaging transducer at the time, and showed some effect on mood. So I know Jay who did this, and I don't count that as the first focused ultrasound study, but maybe one of those details is off, but I don't think so. Okay, but ask again if that's not what you want. So real quick, what is the mechanism of this? So we're able to cause a reversible change in neural activity by putting pressure waves through tissue, through neurons, right? Like what underlies that? So just like really every other form or most other forms of neuromodulation, the mechanism is not fully understood. There's some competing theories here, and that shouldn't scare us because we haven't fully characterized TMS or TDCS either, though we have a pretty good idea. Increasingly, we have a pretty good idea of what's underlying focused ultrasound as well. So back in kind of like the 2017 period, a lot of people were leaning more towards, or certainly people I was associated with, were leaning more towards this first thing shown on the left. This is, listing several theoretical mechanisms of action, or excuse me, not on the far left, but this mechanosensitive ion channels. So it was thought for a while that probably what's going on mostly is that, though it's weird, people have noticed that the mechanosensitive channels that you have in your skin that allow you to feel somatosensation are also expressed in the brain, or at least there's plenty of mechanosensitive channels expressed in the brain for some reason that you couldn't imagine why that is. And it was thought that physically opening these with pressure waves might induce the depolarization and action potentials that we're seeing following focused ultrasound. And there is some animal evidence for that. However, nowadays it's increasingly clear that that's not underlying the majority of the effects, right? You can block these out and still get the effect in animal models. Okay. So without going into too much in the nitty-gritty there, we talked about later, there's been kind of increasing emphasis on this model implicating cavitation or stable cavitation. So when you put pressure waves through a fluid with dissolved gases, which water in the brain inherently has, the negative pressure wave, right, that follows the front of the pressure wave, just inherently can generate gas bubbles if it's of enough negative pressure, right? It's like a vacuum. We pull gases out of the fluid. They form for a brief period of time. What's stable cavitation is that they don't then explode, which can happen at a super high intensity, right? But when the pressure equalizes or the positive pressure wave comes in, they redissolve into the gas. And this is happening, throughout some significant, pressure threshold when putting ultrasound into the brain, including in between the lipid bilayer, and that's what it's showing right here. Yeah, you can see that. So that's what's showing right there. It's thought that the overall effect of this is to create some depolarization in the membrane, and action potentials following that. It's nothing about that model. Like computational models built off of that concept seem to explain the most accurately the available data that we have, and that's my understanding of why that's become the most popular of these models right now. There's a lot of good like bench work on that right now, and so some people have told me like, oh, it's 2, 3 something years out, and we'll really know. So we'll see if that happens. Okay, so that's the majority of the background, and now I'm going to kind of try to do my best to overlay the work that I've done and that some other people have done onto that. So focus ultrasound, right, is able to hit anywhere in the brain pretty accurately, right? Not just in theory, but increasingly really in practice here. And think for a second not only of all the potential scientific applications of this, but of the clinical applications as well, right? So this, I think pretty soon has a very good chance of really blowing open the field of psychiatry, because we can finally directly target all of these deep regions that appear to be particularly implicated in so many conditions, right? The amygdala, for instance, right? Like maybe we can reduce anxiety in anxious people by just directly turning down activity in the amygdala, for instance, as a simple example. Now, okay, I want to give my example of this. What I did my PhD in at UCLA under Martin Monti was built around the idea of using FUS to treat disorders of consciousness patients. So this is coma and related conditions that you can see here. Actually, most of them are not really in coma. It's a misconception. Most people actually don't last in a coma for a very long period of time. It's just like 2 days or to 2 weeks at most. And then they either pass away or will recover into a vegetative state or a minimally conscious state. So these are the types of patients that we are assessing here in this trial. Okay. So I'm gonna speed it up a little bit. Okay. Okay. So, for instance, right? Why use focus ultrasound at all here in this context instead of something else? And it's because these deep brain regions like the thalamus and the basal ganglia are particularly implicated in this condition, right? So this is DOC, disorders of consciousness, are really, really hard to treat. We can keep these people alive, but there's not a lot that we can do to bring them out of their state of reduced consciousness, reduced or absent consciousness. And one reason for this might be because we just can't get to these deep brain structures without putting an electrode in there, and most of these people cannot undergo surgery, right? So an obvious maybe choice is to use this technology. This is just a bunch of evidence for, implicating the deep brain to directly target these regions, and this is what we did, in particular, the central thalamus. Because it is so related to arousal, and there was already deep brain stimulation data suggesting that targeting the central thalamus was able to generate whole brain arousal in these coma patients and their related conditions in these DOC patients already. In fact, you put a DBS electrode into a monkey, wake it up from anesthesia, from sleep. It's pretty compelling. So this was the general idea here. Okay, so this summarizes, actually a number of studies now, and actually the very last one in our last cohort, I'm publishing right now. So many years later, but to generally summarize, in a number of cohorts now, and actually several clinical trials, this data being from the first, we see a consistent behavioral recovery in these patients. It's not, on average, a massive effect size necessarily, because there's also a lot of variability, but it is consistent in both acute patients and chronic patients. So both acute patients that have just had a brain injury and which induce some reduced consciousness, and in chronic patients that may have been in one of these states for years, one of them 20 years, I think was the max, okay? So we show this behavior recovery, right? And we're doing this while aiming our transducer in the MRI, right? So it's MR guided. And of course, if you do that, you might as well collect functional data. And so we have that as well to kind of summarize everything, like a lot of complex data. Instead of the excitation in the central thalamus that we expected and intended to cause, we're seeing inhibition in that brain region and in some of its targets. And this actually is in line with a lot of the data that would then come out in the years after, including my own, suggesting that our parameters are inhibitory. So it's totally to be expected now. It's not what we would do, but we do see this inhibition, right, in the thalamus. What's interesting, though, is that we also see changes in connectivity, both increases and decreases, and these changes in connectivity are associated with recovery in these patients. Kind of complex patterns, but ones that really make sense. I would just really encourage you to read the papers then, for more details. Okay, so before we get to our modern stuff, I'll just say there's been a whole lot of work since then, right? So since that kind of 2013 to 17 period where the really, really early stuff happened, that we just talked about, right? Okay, there's been so many advances since, and here's just a few, and this is getting a little dated now, but even back, right after this period, for instance, this Korean group showed that you could do this very basic BCI using focused ultrasound, where one individual even between two people, right, where one individual would look at a screen, get a signal, it's just like right or left, and then they would intentionally think about performing a behavior, I think touching to the right or to the left, and they have EEG on. That's decoded, sent to someone with this ultrasound helmet on, who gets a stimulation to the somatosensory cortex of one side, and they feel a tingle in the respective hand, and then report apparently well above chance, right? If this person's thinking to the left or to the right, which is a really amazing early study. For example, we can change tactile sensitivity, and then here's one of my studies showing the first bold effect from subcortical focused ultrasound, specifically in the basal ganglia and thalamus, these things related to DOC.
Josh, it's your 5-minute warning, and I think we'll go, we can if you can close it up around the end of the hour, and then we can take some questions for those folks that can hang back. Okay?
Yeah. Okay, sounds good. Yeah. No, I was wondering if we could. Okay, sounds good. So I'll go fast through this. It's always more than I expect. Okay, so I'll go faster through this. So this is like my most recent study, right? So this brings us to the current state of things, right? How is professional focused ultrasound done in the context of science? And at least, arguably, and I hope so, that's represented by my recent study attempting to use it to induce deep meditative states in experienced practitioners in particular. Okay, so basically the justification is pretty simple. People use things like neurofeedback to attempt to induce deep meditation, but there's like this complicated state space you have to move through in order to get to that target state. And there may be these points where activation energy, right, is necessary to get over these humps and get all the way there. Is my argument. There may be some benefit basically to just directly changing brain activity in some neurocorrelates of meditation to get people to those deep states is the idea. So this is the design. So we're hitting these 3 different neurocorrelates, PCC, caudate, insula, for 12 minutes in the middle of a 1 hour long meditation, and comparing those to each other, and then also sham. And an element of this that is particularly reflective of modern techniques with this technology is that it goes all these different modalities. We have physiological methods and such. This is happening outside of the MRI, using neuronavigation, which has become incredibly common. I would encourage people that are interested to look at this. It's just a preprint right now, but it's going to be fully published soon. This is just the first 12 subjects that are experts versus novices. That's online, and we have a whole cohort of 36 people that I am analyzing now. The sham was good, appeared to be effective, and then real quick. Okay, 2 minutes. Okay, good. So real quick, the data I'm going to show right now, which is like the core of this study, is subtraction data. Okay, so this is really simple for a lot of people here, I'm sure, but it's just important to understand. So I'm going to show the difference in our targeted brain regions, right, on a particular measure, okay, over time when subtracting sham out. So if there's any difference from 0, that's interesting here, right? There's like an increased reported depth, right, at a certain point in this data that is compared to sham, and it's interesting. So when you look at our first 12 experts here, these are 3 different targeted regions. Blue is, that should be shown, but it's not. Blue is PCC, orange is the caudate, and yellow is the insula here. Okay, over time. The focus ultrasound, importantly, is turned on at minute 12. So that's like this red dotted line to this green dotted line, okay? And really cool, we see this general arc of an increase in reported depth, meditative depth, for a good chunk of time in these experts after the ultrasound actually is turned off fully, which is not an uncommon thing, for instance, okay? So we see a general effect for some of these of region, and then specifically, if you had to pick one, the caudate in particular seems to particularly increase depth in experts. You don't see anything in the novices, which makes sense. So we train them how to meditate. They know a little bit about what they're doing, but they're probably not perceptive enough to understand, if it's like increased meditative depth is one hypothesis here. And when subtracting them critically, you see, as you would expect, pretty much the same thing you see in the experts alone. Okay, and we're excited to see what happens in our larger cohort of subjects. And then, really quick on that, basically, you reduce heart rate too, increase heart rate variability. and they're super, super correlated, really high within our experts. And then we're going to go on to, I'm going to throw this into the nitty-gritty stuff since it's 7, and the people that want to stay can do that. We'll talk about it because it's too complicated to go into. So, Tiff, I don't know if you want to say something.
Okay, so are you calling it here? This is sort of like, this is a good stopping point?
Well, I'm calling this like, I mean, look, it's 7, right? So if you want to call this the hour and then people can stay, then
yeah, I mean,
yeah.
That's fine. Do you want to continue presenting, or you want to take questions? Are you ready?
Well, let's take questions now. Yeah.
Okay. Well, so if anybody has questions, I mean, it was really fast and intense and dense.
I know, yeah.
Yeah, but it was great. So thank you so much. Thank you so much. Does anybody have any questions right now?
It's probably a lot.
Well, I can ask a question.
Yeah.
So how far are we? Well, actually, let me ask it this way. How much is this being used in healthcare at the moment, and how far are we, do you think, from this becoming mainstream?
Mm-hmm. Yeah, that's a good question. So it's interesting. Most of the very early human stuff was more clinical trials, right? Like really early phase 0 clinical trials. And then just because that process takes so long, it kind of switched back to an emphasis on the science to improve things. And now I think there's been more and more success in some, very forward-thinking psychiatric clinics and in a clinical trial setting. So it's not FDA approved, but it's in that pipeline in some form. I honestly, I don't want to say exactly where, but it maybe is a couple of years out from FDA approval for a couple of disorders, right? Maybe disorders of consciousness, which is, the stuff I started, or for movement disorders like essential tremor, where we're getting some really cool results. And that's actually what I'm going to start doing here as well. So I really feel like this year is the moment. It's kind of like exploding into the mainstream in general, and that is going to be particularly the case the second it gets FDA approved for anything, which is probably in the next 5 years for sure. Yeah.
And forgive me for simplifying this, but is there 2 main modalities in terms of or applications? One being like burning things out, and then the second being a stimulation.
Yeah, you could put it that way. So HIFU is just burning things out. Yeah. And then LIFU, which is one of these, there's like a million acronyms, like it hasn't coalesced onto one of them, but like low intensity focused ultrasound is okay. So it is generally for neuromodulation, but there are some other applications that are gaining interest a lot actually recently. So for instance, you can also inject microbubbles systemically, right? So into the whole body, and use this paired with ultrasound to open up the blood-brain barrier for a period of time, and allow in larger things that normally wouldn't get into the brain, like large drugs, really any drug maybe, and even like cellular components. So a lot of people are getting very interested in that as well. So I'd say those are the main. Yeah, those are the main applications, and there's some other more experimental things.
Right on.
Yeah.
Can I ask a question? About a month or so ago, that 60 Minutes they came out about ultrasound with addictions and Alzheimer's at WVU. So this sounds a little similar. It's a phenomenal presentation. I'm curious, how similar or different that is, or it's just another extension of what you're doing.
Yeah. So a lot of people have seen that, and so it's kind of introduced this to a lot of people. And that's specifically the opening of the blood-brain barrier that I just mentioned. And that's what I mean is kind of gained a lot of interest now, but that's been going on in the background for years. That's definitely been a huge thing, right? So there's so many drugs you just can't introduce to the brain because of this, this layer of cells covering all the vasculature there. Now we can totally open it anywhere in the brain in a particular region for some period of time, and let these drugs in and other things. And that's what they did there to clear out plaques associated with Alzheimer's, using a drug that otherwise you just can't get into the brain. That had been done before in plenty of animal trials. I'm not even sure if, and I think it's in some human trials as well. But just, being on 60 Minutes, a lot of people have seen that. So that's what it was. It was just BBB opening, That's at the same intensities I use. The only difference is that you inject these little tiny microbubbles or nanobubbles that explode and kind of force open little gaps in the blood-brain barrier for a period of time, let these big things in,
I've got a question here. Thank you so much, Josh, by the way. Great.
Oh, Luke, what's up? Hey. Yeah.
So I know that the research is a little bit fresh on this topic, but I was wondering, what are the main factors within a protocol that affect whether or not the TFUS ends up being neuroinhibitory versus stimulatory?
Yeah, that was one of the nitty-gritty things that is it's super important, but it's so complicated to talk about because it's there's all these, it's about 5 or 6 dimensional, the whole space of parameters that are possibly relevant for this, right? It's often we give it in pulses, of course, so we can apply it at certain frequencies on top of the main sound frequency, right? So if you want to do like a theta burst thing, like in TMS, you can do that by applying these pulses, right? That's one thing that might be important, right? Is the frequency of the sound itself important, right? Is it the duty cycle, for instance, which is this other important thing? That's how long it's on in a given time period versus off, right? So it could be on for 5% of the time, right? In pulses, and then off, in between these pulses for 95% of the time. So there's all these things, and that's just a few that could matter. So people are kind of just jump into one part of this n dimension, the 6-dimensional space, and see what's that look like, and so it's sparse, but it's getting more figured out. Basically, the field has coalesced increasingly on the duty cycle being really important for whether it's inhibitory or excitatory. That's been constantly replicated in cortical tissue in particular. And there's this is actually a lot of the underlying theory behind this stable cavitation thing I talked about earlier, the little bubbles, forming in between the lipid bilayer. The models produced from that produce this really cool, actually, maybe I'll find it. These really cool predictions. Huh?
Is that the bilayer soniform model?
It's no, it's actually not. It's really similar. But basically, it's right here. So this produces certain predictions about would be, excitations of activation and suppression in cortical tissue, given a whole bunch of things. It's a really complicated model, and it perfectly describes the data that we had at the time when this came out, and the vast majority of it since, which has made it really popular theory. Basically, in general, at a certain intensity. So if we're gonna say most of the intensities are around here, right? That we would use in humans. This is actually exactly the sort of parameters I used in the work that I showed here, right? If you have a higher duty cycle, especially above 30, it's usually going to be activation in cortex or in the thalamus, which also has been looked at right? If you have a really low duty cycle, it's going to be suppression, but it's unclear how much that applies to other brain regions. Yeah, that weren't modeled. Okay,
Thank
so
you.
that's what. Yeah, I didn't go into that because that's what it just looks like so much, and that's just the tip of the iceberg. Yeah. But it's a general trend.
All right, Josh. Well, that was amazing, and really appreciate finally getting to hear you speak. And I speak for all of us when I say that, but I've been waiting probably the longest. So thank you so
Yeah,
much
I
for
think
your
so.
time and being a part of our guest speaker series.
Yeah. Well, thank you very much, Tiff. Yeah, it's a lot to cover. It's a lot to know if you want to get into that field, as Luke knows, as been forced to do recently. But you know, it's worth it for all the advantages, in my opinion.
I think you're right. Yeah, we're gonna have you back for sure, and we're gonna do it again. So thanks. Have a great day, everybody. Thanks for jumping on. Yeah, we'll see you next time.
Mm-hmm.
Okay, bye.